Okay, well, friends, today we are going to have another very fascinating lecture in the series Hot Talk 44. And friends, you will be happy that we have with us a very renowned microbiologist, Professor Ibrahim Ortas. It's a privilege to have him as a part of Hot Talk 44. And friends, he's a renowned scholar on microbiology research, and he's the man who needs, in fact, no introduction. But before he takes over his presentation, I would like to introduce him to all of you. And I'm sure that you are going to have a very fascinating lecture out of him. He's a man who has spent almost entire life in microbiology research. Right now, Professor Ibrahim Ortas, he is a professor at the University of CUKUROVA, Department of Soil Science and Plant Nutrition. And friends, you'll be happy that he's the man who has spent almost entire life. He has done PhD on mycorrhizal research in sorghum way back in 1994 from the University of Reading, United Kingdom. And thereafter, he joined as an assistant professor, then associate professor and professor. He is the professor from 2010. Sorry, he's a man who is referred word over. Yeah, and you'll be happy to know that he is the member of International Mycorrhizal Association. He's a member of International Union of Soil Science. He's the member of Central Board of Subtropical Fruits. And he has got nearly 81 peer-reviewed ISI publications, and he has published in some of the world's top-class journals, being so widely referred in the journals like Journal of Plant Nutrition, Communication in Soil Science and Plant Analysis, ACTA Horticulture, ACTA Agriculture, Soil Fertility and Plant Biology, European Journal of Horticulture Science, European Journal of Soil Fertility. And he has got nearly 25 chapters, and one of the, I think, the renowned chapter which I remember, which he has contributed into that is his chapter into the Encyclopedia of Soil Science. And friends, it is only the very privileged ones who get the chance to publish into Encyclopedia of Soil Science. And he has got nearly more than 1,000 publications, has contributed immensely towards the betterment of the farmers, the radio talks, print media, electronic media. He's a man, in fact, he's a man of real research, and he's a man of shall I say that he's the best friend of the farmers. So wherever he has been working, he has always looked into the benefit of the farmers. And with this small introduction, may I request our friend, Professor Ibrahim Ortas, to kindly deliver your lecture, please. Professor Ibrahim Ortas, please.
I'm very happy to talk to the very distinguished scientist in horticulture of Indians, and also I know many of Indian scientists since 1998 from the Germany, from UK and United States, and in many places I have been. I have a very good relationship with the Indian, also been visited twice previously in India. Anyway, I'm just going to give some talk about what we are doing in CUKUROVA University and what we have. Sorry, first of all, I should go to the... Is it all right now?
Yes, sir.
Is it all right?
Yeah.
Huh?
Yes, yes.
Okay. So where we are in Anatolia. I am living in Adana here, if you are seeing, and Turkey is in the part of between Asia and Europe, but mainly we are in the Asian sides, and I can tell what we are in the places where mainly the agriculture is one of the oldest agriculture all over the world as far as we know. So what we do, I should say, We are in the places, you know, maybe you heard it, the Göbekli Tepe very recently has been founded. It has a history up to 16,000 years old, and the site of that area, the mountains of the Nemrut, and in the central of Anatolia we have a Cappadocia, around the Izmir we have a Ephesus, and this is Pamukkale, this is Antalya, they are very famous area. This is the city of the orange, and I live in these places. This is the Adana city. It is one of the largest agriculture area cities. So I do work in the CUKUROVA University, and the CUKUROVA University is the one of the big in 10 university all of the Turkey. So in the places we work, we have a biggest area, we call that one is the CUKUROVA area. It means the lowland. It has been established by the two rivers, Seyhan and Ceyhan rivers. So as I said, this is an orange city. Orange blooms festivals is recently getting more and more popular inside of Turkey, and the spring times when the orange they are flowering, and very nice smell of this orange festival. So many people in different parts of the Turkey, they are coming to get them fun. And so what we are doing in this Adana, in this orange city, I am doing charging and the Rhizosphere lab, and I'm in the CUKUROVA University. This is the CUKUROVA University, and a part of our university. This is our faculty. So the major problem in our area, the chemistry of the soil and soil fertility, and the fertilizer management or sustainable productivity, nitrogen cycling and sustainable agriculture, plant nutrition, mainly nitrogen, phosphorus, zinc, and iron, as they are very limited, as in India or many other parts of the world. We do have mycorrhizal study, we have climate change, soil carbon management, and the biochar research. Those, the red colors has been written, I do deal. Soil conservation erosion study is very important for the Turkey. Nearly 70% of the Turkish soils, they are under the erosion. Mathematical modeling and soil survey, mapping, soil classification, evaluation, and mineralogy are the main research area in our department. In the CUKUROVA area, our soils mainly they are alluvial soil and red Mediterranean soil. As you can see, this is the part of our university research land, and you can see some plants. Mainly they are citrus growing area, and people they have a different the cultivation way. And also we have some research farms, and I am charging our research farm. Those are our research experiment, and students are getting some soil samples, and we deal with the many plants, and we do biochar, we made a compost, and this is our lab and my lab. We produce biochar, and we are measuring carbon flux in the soil and under the field condition. And we have several long-term field experiments. I can give you some idea. Again, I should give you some idea about the horticulture of Turkey as been requested, and this is the CUKUROVA University. Again, it's the pictures from the air taken. It's a really very wonderful places located. And Faculty of Agriculture, we have 10 different departments, from animal science to food science and horticulture and plant science, crop science and plant protection, soil science, agriculture, economic, machinery, and irrigation, whatever. This is one of our departments. This is our research farms. In our research farms, we have 2,000 hectare land. It belongs to our Faculty of Agriculture. This is the Department of Soil Science, and I'm charging this area. We have 11 different long-term field experiments in these places: organic farming and phosphorus, long-term tillage, and whatever. This is some pictures in our research area in Adana. So in the area we work, we have a high soil pH, high calcium carbonate content, and heavy clay soil. Soil structure development is a little bit problematic, and we have iron, zinc, and phosphorus deficiency, and in some area we have a salinity and cool and forest as well. And I can tell you the Turkey, when we look at the Turkish maps, we can see there is 7 main ecological regions and 4 transition sections in terms of the agriculture. Mainly we have a Mediterranean climate and semi-arid climate. In the central of Anatolia and east part of Anatolia, it's a little more dry area. As I said, Turkey is climatically, it was a suitable places. And in the Europes, we have more than 12,000 plant species, and Turkey also have 12,000 plant species. Nearly 4,000 plants are endemic plants. And we have 75 fruit species and 60 vegetable species are cultivated. Among the grape species, which is vinifera, and it is 60 variety is cultivated. And usually we have a micro gen pool center. We have 4 centers. One is the Thracia Aegean area, southeast Anatolia. Samsun, Tokat, Amasya is the northern part of the Turkey. Kayseri is in around the middle of the area. In that places, mainly the apple, almond, pear, grape, lentil, cover, all the fruit species. And in the outer area also apples, cherry, melon, legumes, and fodder plants are growing, cultivated mainly. As I said, in the world we have 130 different variety of fruits, and 75 can be cultivated in Turkey, mainly subtropical, tropics, and temperate zone plants. And also we have a vegetable, 200 vegetables, 60 are growing in Turkey. So Turkey, when we look at the Turkish agriculture land, the majority of the land is located for the field crop, nearly 66%, 70%, and we have something like 4 million hectares is for following. For vegetable is around 1 million hectares, and the fruit area is a little bit higher, around 3.2 or 3.3 hectares of land is located for the fruit growth. When we look at the agriculture production, and the production mainly in between from 2000 to 2020, and we yearly produce 68.5 million ton field crops, and we produce 31.3 million for vegetable and 23.5 million ton fruits. Totally we produce 123 million ton yield. In some of the plants, as I said, they are the #1 in Turkey. Hazelnut, cherry, fig. Fig, you know, is the mother of the fig area, and apricot and quince. They are the major crops are growing in Turkey. So cherry, it's really recently getting more important. Chestnut, 75% of the all over the world, chestnut is growing also in the Turkish area. Melon, watermelon, cucumber, chickpea, pistachio, pepper, apple, walnut also, olive, tomato, beans, Spanish, lentils, all are grown. Tea, eggplant, mandarin, onion, grapefruit, lemon, potato, whatever. I mean, many of the plants are growing in the Turkey. And our production in nut, cherry, fig, airport, and quince, and pistachio also is growing in the north part of the Syria and south part of the Turkey. Olive, tomato. They are one of the major crops, and horticulture crops are growing in the Turkish area. And also we have a greenhouse production very recently is getting increased because there is a more demand from the Russia and some other country, especially tomato, pepper, and vegetables. They have a huge demand actually. Whenever again when we look at the area, in many areas recently supposed to be for the forage area, but people they converted into agriculture, mainly they put in the tree plants. So the different form, you can see the different plant growth, and you have a huge, largely nearly 75% of the orange in this area, different variety. As I said, the orange is one of the major plantation in Çukurova part of the coast of the Turkey. Nearly 3,000 species is endemic flower in the Turkey that we can find it. Again, it's one of the important Mediterranean plant, the citrus. Nearly 0.5 million seedlings per year is produced in only in our Çukurova region. You can see the pictures, and from the south part of Turkey, from coast of the Syrian border and Antakya, and in the coast of the Mediterranean, of the Aegean Sea, and small quantity of the area in the Black Sea side around the Riza. This is a microclimate area. We produce citrus in this area. The production is and population, when we compare it, the plant production and the population is increasing. And what I should say in our production of the citrus is year by year is getting increased. It's, as I said, is one of the major one. Mainly is orange, mandarin, lemon, grapefruit, and others. Totally, we produce 4.8 million ton of the citrus. And citrus production, you can see according to FAO data, and it's year by year is increased. I was just trying to look at the production of the citrus in India and Turkey, although India is producing really big size, but Turkey also, they are producing up to 4 million, but India is producing much more. Again, when we compare it, the lemon and mandarin, mainly our production and export is located and concentrated on orange. As I said, the soil fertility and plant nutrition is a problem in our area. And when we say what is the solution, what we can do, because the plant nutrition, since our not only we human, also the other animals and plants are much more dependent on the soil fertility. If the soil fertility, if the soil mineralogy, if the soil body, it is problematic in terms of the plant nutrition, it means your plants have a problem, and it means through the food change, it means you human, you also have a problem, because our food is indirectly or directly it's coming from the soil. So that's why it's very important. In order to avoid the artificial addition of the chemical or fertilizer, some others, we have to have a million years of the evolution of the plant and soil. We can have the mechanism and the plants are producing by themselves. And so far, the plants, they survive and they grow up to present time. So because of our soils, clay and high pH and high lime, and the nutrient availability is very limited. So that's why we have to use a mechanism of rhizosphere. And one of the biggest mechanisms in the rhizosphere, it is mycorrhiza. Mycorrhiza, it is a fungi, and that fungi is a root fungi. Nearly 90% of all over the world's plant, million plant species are mycorrhizal. Some of them, they are highly mycorrhizal demanded, some of them, they are less. Mycorrhizal fungi is the largest symbiosis in between root and the fungi, which is essential part of the healthy plant growth and survival and soil biological quality. And I said the plants are depends on the organism. Some plants are dependent on the rhizobium. Some plants are depending for actinomycin. Some majority, as I said, they depend on the fungi, mycorrhizal fungi. A mycorrhizal fungi is a friendly organism in the soil you can find, and they have a symbiotic relation, interrelation with the plants. They are supporting each other, they are helping each other, and some part is giving the nutrient to the plants, and the plants is supplying carbon to the fungi. So when we look at the plants, as I said a few minutes ago, some plants are very strongly mycorrhizal dependent. Who is those plants? Those plants are mainly, they are horticulture crops. For example, avocado, black pepper, cassava, citrus, coffee, cacao, coffee, guana, mango, linseed, alpha bean, all are pecan pea. They are strongly mycorrhizal dependent. What does it mean strongly mycorrhizal dependent? It means even you are putting more fertilizer, the plants again is demanding the mycorrhizal. Because the mycorrhiza is not only getting the phosphorus, also getting some other nutrient and water as well. And also it's make a change in the physiology of the plant, and it's helping to plant to get resistance against the biotic, abiotic stress factors as well. So some plants are very highly dependent, some are high, some are low, something like a canola, cabbage, brassica guruva, broccoli, spinach, and radish, they are not mycorrhizal. As we know, the million and million plants in the soil, they get the nutrient, but each have their own rhizosphere mechanism to get the nutrient from the soil. So it's not easy. We are challenging. We are working to get them food, and also plants are also getting challenged. It is spending some energy to get the nutrient. It's not easy. Plants are really challenging to take up nutrients from the soil surface. So that's why each plant have their mechanism. Excuse me. In this way, when we look at the highly dependent plant dependency, it's mainly depend on the root development. I mean, the root surface area is very important. The root system surface area, branching frequency, root hairs, root activity, root growth rate, responsibility, soil condition, primary root lifespan, and root exudation. They are very important, and the mycorrhizal formation. Those are depend on whether the plants are mycorrhizal, or the dependency of degree is how is changed. It depends on the root parameters. Root parameters shouldn't be neglected by the soil scientists. Actually, I can say, if you are a good growers, it means you should be a good rhizosphere manager. If you know how to manage your rhizosphere, it means you're applying organic manure, you're applying all the fertilizer, your management of the soil cultivation or whatever, all are directly or indirectly related with the root growth. And you change the root area and you change the nutrient availability. Don't forget it. Not only the scientists, also the farmers, they should be aware of this root area management. So some plant, they are giving a different response to the mycorrhiza. For example, in citrus, kiwi, and apple plant, when you inoculate it with the mycorrhiza, you stimulate the plant growth. For example, citrus, you can increase the yield, and also you can increase the plant resistance against the water deficiency. For example, for grape, and the sweetness is increased. For the banana, the plant resistance against the disease is increased. Against citrus plants is challenging with the drought tolerance. So how is it's happening this one? When you look at this one, when I say the rhizosphere is in this area. In this area, you can see there is no root hairs. There is the limited of rhizosphere. If you have a root hairs, you have a little bit a rhizosphere zone. But if you have a mycorrhiza, you have a very large expanded area. I mean, if you have a only root, your roots are getting nutrient from this area. If you have a root hairs, the plants are absorbing nutrient from this area. But if you have a mycorrhiza and mycorrhizal area is in a large area, the hyphae are influencing or penetrating inside the soil, and they get a nutrient and the waters. So that's why the rhizosphere expandation is done by the mycorrhizae. Mycorrhizae is increasing also the surface area, because 1 centimeter of the roots, they have more than 100 meters of root hyphae. When you convert it into the surface, make a simple calculation, and you will find really a large area you produce to touch with the soil and to get the nutrient and the water from the soil to inside the plants. So mycorrhizae increase the nutrient mainly in the phosphorus. The phosphorus mechanism, we know, it is very complex. So many root solution and soil solution, and also the macroorganism solution and the hyphae and surface, so many parameters are involved in this area. Then you can see that these pictures, and some of them I draw by myself, and you can see in the rhizosphere area, as I said, and a large area. This is the mycorrhizal, and this is only the rhizosphere root hairs, and this surface is only the root surface. And the Marshner and the group, they did some work. If they have a mycorrhiza up to 20 millimeter from the root surface, you expand it, you depleted the nutrient. But if you have no mycorrhiza, in just a 5 millimeters from the root surface, you depleted the nutrient and phosphorus and zinc and copper. But the mycorrhiza is really depleted nutrient from the surface, but the roots is not doing the same. So what is the magic behind? The magic is, this is the hyphae diameters, this is the root diameters. You can see this. When you look at around the root under the microscope, you can see a million hairs, hyphae, like our hairs, they are around. In the rhizosphere also, you can see again the pH is changed by the nitrogen nutrition. And I drew this one in my PhD, and you can see on the surface rhizosphere pH soil is different than a little bit outside of the rhizosphere area. And they make a calculation up to 3 units of the rhizosphere change. It is done by the management. What I was saying, if you are able to change or manage your rhizosphere by using your fertilizer, whether you use the ammonium or nitrate, you can change your rhizosphere. You have to know your soil. You have to know your soil characteristic, your soil pH, your soil calcium carbon, and also which type of plants, and if you are going to cultivate it, and what is your plant's nitrogen and ammonium demand, nitrogen demand. That's are very important, because you are actually managing your rhizosphere. That has been always done. And also we know the nutrient, some nutrients are interceptually taken by the plants. Some they are coming from with the diffusion, some are coming with the must follow with the water. So that's why again we have to act consciously how we can manage our soil, how we can manage our crop and soil management system to get a better response. So what we do in our lab, we have two labs in my groups, a rhizosphere and soil biotechnology lab. We said that we are focusing on whether or not the mycorrhizal formation in the several field crops and forestry plants, and also we are trying to look at once the mycorrhiza do form properly in the plant roots, what are the consequences of the host survival and growth and nutrient uptake, and management of the identified field mycorrhizal effect on the plant growth and soil quality. I will just give you some example. And the indigenous mycorrhiza, I know I read it several people and Indian scientists, and also they get some good response. So those are our focus major on the area. So since 1995, I work in the department, and we, in the beginning, we produced 2 strategies. Recently, we increased to make a 4th strategy for our lab. The first one is using selected mycorrhizal fungi for the field crop and horticulture crop. You know, how we can mycorrhize or whether we can use or not. The second one is using mycorrhiza for field crop is really very difficult. You cannot apply, say, something like an 400 kilogram, 500 kilogram mycorrhizal inoculant, because we grow in the still, we have a problem in production of the mycorrhiza. But it is better to use the mycorrhiza for field, not for field crop, for the horticulture crops, because the horticulture crops mainly they are producing from the seedling stages and then they transplant it to the field. So it is easy to use mycorrhizal inoculated seedlings. And we did quite lots of work. I'm going to just show you. And also we have a soil and crop management system. We have some long-term field experiments. Since I'm in the departments, in the 1996, we started some long-term field experiment. And so far we have 11 different long-term field experiment, and we are challenging also to look at the soil management, how is influenced the indigenous mycorrhizae in the soil, and they have a consequence on the soil carbon and climate change and soil development as well. But the fourth one is a soil mycorrhizae, soil, plant, and atmosphere relationship and related with the carbon. So what we did, we did quite lots of work. I'm just going to pass this one. So far, we study more than 30 different plants: wheat, garlic, tomato, pepper, eggplant, melon, onion, horsebean, marrow, apple, olive, pistachio, banana, grapevine, persimmon, apricot, fig, cherry, citrus, soybean, potato, whatever. I mean, we work in quite lots of plants, and we just try to look at which type of mycorrhiza is working, and the plant species, they have any preference of the mycorrhiza. And so we try to also to understand, you know, how is acting in a different perspective. Is it all right? Anop?
Yeah.
It's all right?
Perfect, perfect. Go on.
Okay,
Go
good.
on, and there are a lot of researchers. Give some researchable issues also to them.
Okay, okay,
Yeah.
okay. I mean, somebody else, they can ask the question, whatever
Please,
they
please,
need.
carry on. Perfect.
Okay, okay. So, what I should say, you know, and what we have, and those many plants, we check, we use the different mycorrhizal species, and we try to also look at the growth medium and the plant related with the different mycorrhiza, how they interacted with each other. So what we work also to produce seedling. Seedling is very important, especially if you are working in horticulture. As I said, many horticulture crops are growing seed or seedling in the nursery condition, and then they transplant it to the field. So it's a good opportunity for the mycorrhizal scientists. So we found it, you know, in some plants are up to 90% they get a mycorrhizal colonization. Some plants they get in something like 20, 40. It's very still small. Spore effectiveness is also important for us. Number of the propagal, how much propagal we need to apply, still we don't know. I mean, this the mycorrhizal scientists still, I mean, last 50, 60 years we are working and we produce quite lots of data, but still we have some big lack about the effectiveness and the number of the propagal and which species, which plant species, and each is related with which mycorrhizal species, because we have many mycorrhizal taxa, and also we have a subtaxa as well. So that's why it's important for us for re-inoculation of mycorrhizal inoculum during the seedling transplanting to the field condition is still not well known. I mean, also in this Middle East area and Turkey, less people they deal with the mycorrhiza. Some people they deal in Israel and and Jordan and Egypt, and also I am also recently working in the mycorrhiza in this area. When I look at there is a few in working in Iran, but the other type only in India and China people are extensively working mycorrhiza, but the other part of the Turkish states they are not working too much. So what we did, I should give you some small what we did in our lab and our field, and how we used the technique of the mycorrhiza to the greenhouse and in the field condition. So we use two different, first I try to look at the leek and onion. They are very mycorrhizal dependent plants, and how we can get the better mycorrhizal spores and how much spores we need to apply. What we did, we have a control and 500 and 1200 spores, and more or less, as you can see, look at for leek and onion, for both of them, Glamus mosea and Glamus ethnicatinum were used. You can see, 500, it seems to be okay. 500 and 1000, 2000 doesn't make too much big. And in here, you can see 1000 and 2000 spores seems to be okay. When we try to look at the number of the spores, we found it for the leek and onion, both of them, they are really very good to produce mycorrhizal spores. Some of them, of course, they are different. For the leek, it seems to be producing a little bit more spores. Mycorrhizal inoculated seedling production, as I said, it is very important because in CUKUROVA region, we are one of the larger seedling producers area. The climate is very optimum. Even the winters, it's not so much cool, and people are getting benefit from the energy supply. When we look at the pepper plant, as I said, the pepper plant is one of the highly mycorrhizal dependent plant, and you get a good response. What we did in this experiment, we use in the seed stages mycorrhizal inoculation, and also we inoculated with the seedling stages. You can see in the seed stages and the seedling stages inoculation of the mycorrhiza, both of them, they are working. But sometimes if your mycorrhizal quantity or effectiveness is very low, maybe you cannot get the better response to the application or get a better response. But if your inoculum effectiveness is good and the propagule number, as I said, 500 or 1000 or in between, maybe if they are not very effective, you may not get a good response. For example, here's ethnocotyniums, and when you look at the cocktail, it seems they are not very efficient. But in the seedling stages, again, we can see they seems to be better. But in any cases, calarium, and it's good, and Glamus mosea in the both stages, I think. This is eggplant. Eggplant also I saw in India also you grow up this one very largely. Again, in the seed stages and seedling stages, you can see in the seedling stages it seems to be a little bit much better, because maybe the plants they grow up and they get some expanded the roots root volume area, so giving a better response. But in these stages, when you apply it in the seed stages, because at the beginning the fungi, when they inoculated in the roots, they demanding high amount of carbon from the photosynthesis produced byproduct. So that's why maybe we can see at the beginning a little bit plants they have a little bit losing in the yield, dry matter yields, because the much carbohydrate produced by photosynthesis is leakage to the rhizosphere for making more hyphae and more roots. So this is the tomato also, again, for the seed stages and seedling stages. Again, here we found that seedling stages is good. I mean, under the field condition, mycorrhizal inoculation, it is very important. We produce in a different way. We produce seedlings, and look at this one. We produce different containers and different way of producing, and also we produce in the surface. We produce in the surface and the field condition. We can produce seedlings, and as farmers are hundreds and hundreds of years, they are doing this kind of technique. So what we did, we produce seedlings. You can see in a different mycorrhizal species, and then we transplanted. First we produce from here nested, and then we transplanted to the field and transplanted to the big containers, and then we changed it. And then we went to field. You can see the field condition. You can see the workers are opening the field, and you can just transplanting your seedlings. It's easy. Or I can show you like this. This lady is doing. This is my hand. The first thing I was trying to apply to mycorrhizal. Look at a small quantity of mycorrhizal. This is my seedlings I produce in outside. So we think we can reduce the fertilizer inputs by using alternative way of the mycorrhiza. It is possible, and we can get a good response. I will show you some data. We really get good response, because fertilizer getting very, very expensive very recently. I don't know in India, but in Turkey very recently, because of the fertilizer is produced by the petrol, and we are depends on the outside of the petrol. So if the petrol prices is getting raising up, and very recently Turkish currency is getting loose against the dollars, and we get the petrol through the dollars, and the petrol price is going up, and the Turkish currency is coming down, and the prices of petrol is getting very expensive, and consequently the fertilizer price is so expensive. Whenever there is such a kind of prices increases, the farmers are not able to use much fertilizer. And those years, following those years, in the many plants, we found that the yield, the total yield of the production is coming down. So that's why in those conditions and those days, we can use the mycorrhizal. I'm really very confident for some plants. For example, I will show you some data about the citrus and peppers or whatever we did. I'm 100% sure even the small quantity of mycorrhiza is definitely change the plant growth. So in the field condition, look at mycorrhizal and non-mycorrhizal. This is the tomato peppers. This is the eggplant. You can see, the mycorrhizal plant and non-mycorrhizal, mycorrhizal plant and mycorrhizal plant and non-mycorrhizal plant. You can see, there is a differences. We found it. And 3 years this experiment was done under the field condition. You can see, look at the mycorrhizal and non-mycorrhizal. Look at the real field work we did. So we calculated 3 years this experiment with and without, and fumigated and without fumigation of the land, and we found that the mycorrhizal dependency. So really, under the less fertilizer condition, mycorrhiza is working. It's working, especially in our climate condition. The temperature is high, water deficiency, soil phosphorus level is low, the zinc level is low, calcium carbonate and pH is high. So it means the nutrient availability is limited. Mycorrhiza is able to get the nutrient from the soil much better than roots. The roots itself, it's not able to get the nutrient from the soil. They definitely need an assistant. Not a very small and very weak assistant. They need a strong assistant. That one is the mycorrhiza. So what we do again, we produce seedlings, and the seedlings are transplanting to the field, you can see, and here. And I use different technique. This is my technique. What I did, I was using in the previous experiment, as you see, I was just using the small quantity of granular mycorrhizal inoculum. But in these cases, we thought, it is better. Sometimes the plant roots, they may not attach it to the mycorrhiza. So what we try to do, we make a soup, a solution. When you get any seedlings, just immerse inside the solution and hold the roots is attached to the mycorrhiza, touch it. When you come to here, you can also inoculate it some granular if you like. If not, you can just put it a little bit. You can see. And in these cases, what we found it, pepper plant and watermelon, and this is the Glomus mossia, this is the cocktail mycorrhiza, you can see, this is the control, and this is the mycorrhiza. Mycorrhizal, solution, and granular, non-mycorrhizal, and mycorrhizal, non-mycorrhizal solution, you can see, the mycorrhiza is always giving response. Look at the data has been done with the two different mycorrhiza. So I can definitely say if you use a right mycorrhiza species, right using technology, and right using the quantity of the mycorrhiza spores, you will definitely get a response. And we use different technique for the tomato plant in 2016. You can see control. And all the mycorrhiza and gel solution, seed stages, and we can use the different techniques, and you can see the plants are giving response. When you look at the root colonization, and when you use the gel, the solution, the root colonization also is increased. When you look at the different mycorrhiza, you can see indigenous mycorrhiza working, Glomus mossia is working, they are giving compared to control. When we look at the watermelon is one of the major crops in our area, really giving high response. And different mycorrhiza species, they have a different response. And we did several experiments. It's a good way to inoculate mycorrhizal, inoculated watermelon seedlings, the beginning. It's a good marketing, actually. So another experiment I was saying, and we are dealing with soil and crop management system under the long-term field experiment. We have different experimental techniques, and the field condition, this is controlled organic manure, mycorrhiza, and mycorrhiza. We use several experiments. In this experiment, this long-term experiment has been settled down in 1996. We have a control, we have mineral fertilizer, we have animal manure, we have a compost, we have compost plus mycorrhizal inoculation. We have 5 treatments since 1996. It is coming on, and we did quite lots of data. We have plenty of data. Some of them has been published, but still we have there's some continuity of the experiment. Excuse me. So, in those stages, we also try to look at the mortality of seedlings. Because, this is very expensive. Very recently, the kilogram of the tomato seeds is equal to cost of gold. You understand? 1 kilogram of seeds is equal to 1 kilogram of gold. It's very expensive. So what the seedlings the people are doing, they are not using seed to the field directly. They use the seedling, and each seed is very important. And the technology also developed, even for the maize, is very expensive. And they are using one by one. So that's why when you transplanted your seedlings to the field, in most of the cases, you lose quite a lot. In terms of the knowledge of the rhizosphere, when the farmers, the plant, macro-propagated plants, I'm going to talk a little, giving some information about that one as well. Most of the plants are growing in the nursery stages, especially under the plant macro-propagation techniques, and you use the seedlings in the sterile condition. But when you go to the outside and the field, there is another life, and that life, there is a different type of the organism. They are directly attacking to the plant roots. The challenge is started. So most of the times, the plants seedlings are not plantless, are not able to survive. So what we did, I just make a calculation. And different plants, tomato, pepper, eggplant, bell pepper, marrow, cucumber, melon, watermelon. I mean, those are the number per plots. You know, it depends on the differences between on line and in two line. And what we look at, for example, the tomato, in the parcel you have 48 plants and one has been died. Number of the death is 1 and 2% it's around, and the 98 has been survived. Pepper, and from 64 has been died. Eggplant 48, 3. Bell pepper from 60 seedlings, 4 has been died. And watermelon, look at 7 has been died from 24. Nearly 1/3 has been died. So when we look at the make a calculation, but in terms of the mycorrhiza, more or less 2 in the watermelon has been died, 1 is in cucumber, the other one nearly more or less they survive. So when we look at this, the survive percentage, you can see the mycorrhiza inoculated is very successfully survive. But in terms of the non-mycorrhizal stages, especially marrow, cucumber, melon, those are the cucumber family plants. They are very sensitive. But compared to Solanaceae familia, in Solanaceae familia, they are much more survivor than cucumber family. So that's very important for us. We check also again another work. We found it again mycorrhizal inoculation. Look at this is the control, Glamosea, Glamosea. You can see the mycorrhizal inoculation that's helping plant to survive more. Actually, you can see the mycorrhiza is a powerful mechanism. It's a really powerful mechanism, helping plant to resist, to be strong, and to challenge with the biotic and abiotic stress factors, and also is protect the plant against the disease. So horticulture plants, as I said, they are mycorrhizal dependent, and this under the macro propagation, mycorrhiza can help the plant to survive the losses and increase the productivity. In vitro and other conditions, mycorrhiza always is helping. So micropropagation, it is recently getting more and more. So many biotechnology technology are using it. We are just using the small plantlets and then transplanting in 1 tubes and then transplanting into the small container from 1 stage, second stage, third stage, and the 4th stage. We are just transplanting to the field condition. So in these stages, step by step, we are trying to make it familiar with the situation. But we shouldn't forget it. In the in vitro and the micropropagated techniques, we avoided some things. No rhizosphere organism. But at the textbooks and the reality, we know the plant without the organism, they cannot survive. So that's why at the beginning, people are dealing with the biotechnology, macropropagation, they forget it, the microorganism. Recently they realize, yes, they need it. Not only the mycorrhizas, the other microorganisms, we still do not know. But we know definitely they have some positive symbiosis relationship. So that's why we shouldn't neglect it. Not only the mycorrhiza, the other organisms, bacteria, fungi, actinorrhiza, whatever. And Victoria, a friend of mine, she from Spain, they transplanted mycorrhizal vineyards plant to the field, and they get this kind of response. And they really get a response, and it is working. Also, we work this kind of vineyard and pistachio, permisia, seedling production. You can see the different mycorrhizal species. They have a different response to the persimmon plants, and this one publish it. And we use again different variety of vitivener and with the different growth medium. That's how we work this kind of thing, because always the plant's growth medium is always very critical. That's one also is related with the roots. If the roots are happy, no problem. If the roots are not so happy, always there is a problem. So that's why growth medium is very important. The nutrient content of the growth medium also is important. You can see we use the different growth medium, and we get a different response. We use the banana. This one also has been published, and we use the banana under the light soil and heavy soil and the mix of different material, and we found that the root growth also is changing. And growth mediums are really different from each other. Look at the control and caladionium and the macropropagation, growth medium 1, growth medium 2. They are different. So that's why for each plant you should know which type of growth medium the plants are getting better growth. And also a different response of the different mycorrhizae. The mycorrhizal colonization is changed. We use the pistachio. Pistachio, you know, is one of the special plants, and it's growing only in the north part of Syria and south part of Turkey. It's a very narrow line. In that area, it's a dry area. The precipitation is around 200 millimeters, something like that. It's during the winter is cool. The growing area is a chalky, high calcium carbonates, but these plants, the sheltered seed plants, is growing differently. We check it different mycorrhiza, and we check it with the different variety of pistachio, Atlantica. And we found that some mycorrhiza are getting better. Glamis ethinocotyledonum, Glamis clarium, they give high response. And the mycorrhizal dependency, also mycorrhizal infection, also is changed. And the biggest work we do so far, we work with the citrus. As I said, citrus is the major plantation in Adana, in the south part of Turkey, in the coastal Mediterranean. 75% of the Turkish citrus production is done in the south, mainly in Adana, in our area. So as I said, nearly 500,000 of the seedlings are produced. In our university also, we have a centers, they are producing seedlings. But we know how people are producing seedlings. They are using some turf, and they get some coconut material from India and different material. They get some turf from Finland, and they just given a huge quantity of the fertilizer, and they produce a good seedlings. When the seedlings transplanting to the field condition, and the farmers are said, all plants are not growing very well. What is the problem? And then they're fighting to each other. And very recently, I mean, more than 10 years ago, when there was such kind of discussion, I said, don't fight to each other, because the problem is in a different way. When I told them, okay, they said, can you help us? Okay, I can help you. But you can see here, you can produce a different way, producing seedlings. Okay, no problem. When you transplant them into the, here, your soil has been sterilized, and you have Glammus mossia and Glammus clarium. They are really different. When you put the same seedlings, and this is controlled, this is indigenous mycorrhizae, this is Glammus mossia, Caledonium, clarium, and this is the cocktail. Cocktail probably seems to be very close to this one. When you look at this one, you can see different, you produce quite lots of seedlings in the nursery condition. When you put in here, look at this one, and when you sterilize the soil, citrus plants is very strange. After certain times, they are not growing if we didn't give anything. But indigenous mycorrhizae is growing in these stages. It means the soil have an indigenous mycorrhizae is making help. But when you re-inoculate a selected mycorrhizae, clarium, we found it is one of the best inoculum. It's working in our area. We get really good response. You can see, you know, how big. This is control. The plant nearly died, but this is indigenous, and this is the Glammus clarium. And also we collected indigenous soil mycorrhizae under the salty area. You can see this is control. This is the mycorrhizae spores we collected under the different the plant species. Look at, they are different, but the seedlings at the beginning was the same. So it means each seedlings, they get a different rhizosphere mycorrhizae, because each plant's rhizosphere is different than the other one. This work also has been published, and you can see, and we look at the different rootstocks, how they respond. Because, you know, in our soil, since we have a high calcium carbonate and high pH, and we have also tristeza disease virus against the citrus plantation, and people are always change their rootstocks. So far we use the sour orange in our system. Sometimes we use the carrizo, we use the also how could you say tree leaves for this kind of work. And when we look at this one, we found it the different rootstocks, they are giving different response to the different mycorrhizal inoculation. Look at the fasciculatum, macrocardium, clarium, and the intraradices. So what we did also, we did quite lots of work with the indigenous mycorrhizae compared to the selected mycorrhizae, because my belief, my rhizosphere idea, each plant, even each variety, they have their own rhizosphere, and each rhizosphere, it has been adapted to that soil and climate condition. For example, I mean, the same lemon, it's growing in India, it's growing in the Turkey as well. But if you like, you can get them a little bit rhizosphere in that plant's roots area, some rhizosphere in this area, and inoculated with the same seeds, you will get a different response. I'm sure, 100%. Because each one, the rhizosphere organism, they are different. Quantity is different, effectiveness is different, and also the inoculation infection point also is different. So that's why I always do believe any organism is in any climate condition, we always should work with them. It is not good to go to get some mycorrhiza from UK or from France or America or whatever. No, I think we can get all regions, rhizosphere organism, we can extract it, isolate it, and we can identify all mycorrhiza, and we can re-inoculate it. This is, I think, it's the best technology. You can say that it is the mycorrhizal technology or indigenous mycorrhizal technology, whatever you say, I think is the one of the best way. So we did quite lots of work. We sterilized the soil without sterilization to understand how the indigenous mycorrhiza are working or not. They're definitely working. Look at, I will show you. Look this one. This is a sterile soil and no mycorrhiza. This is Glomus calarium. This is the indigenous mycorrhiza. When you come here, this is not sterile soil. This is sterile soil. Look at this one. In here, in sterile soil, the plants are not growing, but here is growing. This is calarium. This is the reinoculation of indigenous mycorrhiza. When you sterilize, look at this one and look at this one. Probably, not only mycorrhiza, maybe there is some other organism. Also, they are dealing with this process. So we should understand what is this one. When we look at this one, a non-sterile, sterile condition, when we look at the mycorrhizal dependency, nearly 99% is the mycorrhizal dependency. When we use the different level of phosphorus and different level of the zinc, and we found it again, you know, mycorrhizal and non-mycorrhizal, they are strongly different from each other. And we found that when we increase the phosphorus, mycorrhizal dependency is coming down. Not too much related with the zinc, but too much related with the phosphorus. I mean, we know last 40, 50 years and maybe 500,000 of the papers has been done, and more or less everybody is agree. Mycorrhiza has an affinity with very close relation with the phosphorus. So when you increase the phosphorus, the mycorrhiza is coming down. It means the plant says, if I get the phosphorus, I don't need it another assistant, as I said, assistant. If there is a phosphorus less, I am not able to get the phosphorus from the soil, I need an assistant. That's one, it's mycorrhiza. Again, in this experiment, when we look at the Glomus calarium and indigenous mycorrhiza, we can see Glomus calarium. In this sterile condition and non-sterile condition, you can see when you sterilize the soil, when you eliminated other organisms and re-inoculated indigenous mycorrhiza, and when you inoculated with the Glomus calarium, and you get a high response compared to the non-sterile condition. In another experiment, under the greenhouse condition, we use different level of phosphorus with the mycorrhiza, and if we have no mycorrhiza, and the phosphorus even doesn't make any sense. You remember I was saying the mycorrhizal dependency for some plants, it is an explanation of even you increase the nutrient, doesn't make sense. Plant, in any cases, they needed mycorrhiza. That's this one. This is mycorrhizal and different level of phosphorus, but no response, nearly they are dying. But these soils has been sterilized and we didn't edit anything. But in these cases, again, 0 and 400, mycorrhizal and non-mycorrhizal. When we look at the mycorrhizal plant growth and shoot and root growth, and when we look at the when we look at the zinc and phosphorus uptake, you can see, you know, they are ready with this, and the mycorrhizal dependency, and the 0 phosphor 92, 93, 89, 87, 86. I mean, when you increase the phosphorus, the mycorrhizal dependency is coming down. We have also some long-term field experiment. I inoculated lemon, mandarin, and orange plants with a different mycorrhizal species. My 3 plants are nearly 15 years old. I will give an explanation. We produce seedlings here and transplanting to the field, you know. You can see again I make a solution and I'm transplanting to the field pole. And also I re-inoculated another again mycorrhizal. You can see under the field condition. This is my seedling transplanting. I set up several experiments actually. Still we are working, and also recently we are trying to measure carbon dioxide. And you can see the yield, you know, from the 2014 and from 15, you can see the yield increases. Different mycorrhizal giving different response. You know, the root colonization. So what we have, as I said, we have banana, 1 year, cherry, citrus, fig plant, and pistachio. We use quite lots of different plants and with the different mycorrhizal species. And we found that some plants are really mycorrhizal dependency. For example, here, who is this one? Citrus. Who is this one? Pistachio. Who is this one? Fig. 1 year. I mean, different plants, they have a different response to the mycorrhizal dependency, but the citrus is the one of the leading. So what we have, we found that the indigenous mycorrhizas are really working for the citrus plants, and we found that different mycorrhiza, they are strongly infected some plants under the field condition. Under the field condition, we have the indigenous mycorrhiza. The indigenous mycorrhiza still is working, as they are very strong. And some species, for example, Glamus clarium is one of the significant mycorrhiza for the citrus. We found it. Without inoculation, citrus plants are not given response to the plant. Finally, the results shown that mycorrhizal inoculated plants have a higher yield and nutrient concentration than non-inoculated one. Selected mycorrhiza species and the indigenous mycorrhiza. And I can say the indigenous mycorrhiza or the management of the indigenous mycorrhiza are very effective. It is better to manage indigenous mycorrhiza or to select an efficient indigenous mycorrhiza. And generally, horticulture plants are more mycorrhizal dependent. Again, horticulture plants are more mycorrhizal dependent. Under the field condition, many plants depend on the mycorrhizal inoculation, but also depend on the P supply, P level. It's very important. With high P application, the mycorrhizal dependency effectiveness is coming down. Yes, hand in hand. Thank you very much. I hope it has been clear. It can be discussed.
Great, Professor Ortas. We have thoroughly enjoyed your presentation. Before we wind up, I just wanted to flag off. Am I audible to you?
Yes, sir. Carry on, sir.
Yeah, okay. Professor Ortas, before I wind up, I just wanted to float some of the questions. It's mostly from the audience. The first question which I wanted to flag off, is there any mass scale multiplication of mycorrhiza technology that you have developed so that our researchers also can try it in India?
Why not? Yeah, you can do also. It's not so difficult. If I did something,
okay,
Anop can do 100 times more better than me.
Okay. The second question, Professor Ortas, was that we have indigenous mycorrhiza in the rhizosphere zone of almost all the horticulture crops. is there any technique to trigger off the multiplication of that indigenous mycorrhizas with some treatment, something like that, so that we are able to optimize the activity of mycorrhiza,
Yes, yes,
indigenous
yes.
mycorrhiza within the
What you
rhizosphere
can
of plant?
do, you can first get your rhizosphere soil and the plant roots, and that small quantity of soil, you can make a trap cultures. Some plants are making really good mycorrhiza, something leek and sorghum and maize and onion, and sometimes alfalfa, clovers, and you can use this kind of plants to as a host plant. You can increase the indigenous mycorrhiza. And the second stage is you can get extracted those mycorrhiza spores again. You can recultures again. You can make a better multiplication of the organism, and then you can separate it from by shape, by colors or whatever, and then you can study. It is possible. I mean, we can just make some collaboration. We can do.
Okay. Another thing was, Professor Ortas, we keep talking about the consortium of microbes. Something like that, we haven't seen much headway being made in order to develop some kind of consortium, the cocktail of mycorrhizal. So that instead of one mycorrhiza, you have a two, three species together, and we are able to get the response for a much extended period. And plants are able to address both biotic and abiotic states. Why we are failing on this?
Actually, we did. We use make a different variety of the combination of the inoculum mixing. Actually, what I should say, some organisms really they are working together with each other. Some they are not working. Still, we don't know. But something I know, like me and you, we are good friends. Whenever I'm in international area, when we saw each other, we are just saying, "Hi, how are you?" And we are coming together. And there is somebody else in another side, you know him in another meetings, and you say, "Hi," just pass away. But there is somebody when you see, and then you want to fight to each other, you know? Like organisms, they are like this. Some of them, they like each other. Some of them, they are not like each other. So what should I say?
That's the biggest art to
That's the biggest. That's the biggest art to
Say again?
identify.
That's the biggest art to identify.
That's the
course.
biggest art to identify.
Of course.
Who is
Of
my
course,
real friend?
yeah. So it is very good point. But in indigenous condition, there is so many microorganisms. They are side by side. They know each other. You know, if they are fighting again, they are know each other, but we don't know about them. But they know each other. Sometimes, as you see in my presentation, and usually we put the indigenous microbes. Sometimes they are not working. But, for example, if the same microorganism, they are coming in the same soil, they are working for citrus, but they are not working for the tomato. So, what the question mark is, why? Do the plant, they have a special selection? Or the plant, they have a special preparation for some microorganism? Or this is only the mycorrhiza or some other organism? For example, bacteria and fungi, they are working for some plants. For example, nearly more or less all the rhizobium, the forage plants, they are getting nitrogen fixing bacteria, but most of them, because of the nitrogen phosphorus balance, they need mycorrhiza as well. They work together. And some plants, again, the fungi and bacteria, they work together. But for some plants, they're not working. So still, our knowledge is not sufficient for this kind of work, my friend. I don't know, it is maybe some of the audience, they can help or they can make a discussion for this point.
Okay, Professor.
Okay, Professor. The other thing, you have got such excellent work on mycorrhizal research. I feel many of our friends who are young friends, those who are doing MSc and PhD, they want to take up maybe postdoc, maybe PhD in your laboratory under your leadership. Is there any provision that Indian scholars can really choose Cukurova University and have access to your work and get an opportunity to work with you? I think that is the biggest merit of this lectures today, and I just wanted to
Definitely,
really explore that
but
possibility.
what we can do, or Turkish TÜBİTAK, sometimes they are giving scholars so many Pakistani students. So far, I get it for they come to our lab, and also the Turkish president is giving some. I mean, the office of the presidency, they are giving some scholarship for the students. Most of the African students, they are coming, and Middle East, Indian, I haven't heard, but they can apply. I will pass this.
Why?
Why that happens?
Why? Why that happens? The flow of Indian
Yeah,
students is so
I
less.
mean, it's open for everyone. In most of the people, they are coming from Azerbaijan and Georgia, and they get some scholarship. And I will ask the or foreign office service. I can pass that kind of.
Kindly let them share your notification with ICR, with the state universities in India, so that our Indian scholars also get an opportunity, to get an
Yes,
access to
it is
such a good
possible.
work,
Yeah.
and they
Yes,
can be as good as anyone.
we can make this kind of arrangement. I will be very happy. I mean, it is also same for all students.
Yeah.
I mean, my groups also, they are watching this presentation. We can do this kind of collaboration. I will be very happy.
And Professor Ortas,
And
scholars are the
and
best
Professor
scholars.
Ortas, Indian scholars are
Yeah,
the best world over.
definitely. I mean, we are living in a small planet, you know. It is three and a half hours by flight, but
Okay.
at the moment, because of the COVID-19, we are not able to. But 3.5 hours from India to Turkey, no problem.
You can individually also you can share at
You
least
can
with some of
individually
the
also you
scholars
can
that
share at least with some of the scholars that in India. We can really invite our own students to have some access with you, and they can
Sometimes
have
I get, we
the
know there
PhD
is so many
program.
brilliant students of Indians. 2 years ago, there was an Indian professor. He came to our lab, and I remember, yes, he came to another lab, but he was in the food science, and he said, my background is the soil science. I said, what you are doing there, come to my lab. And sometimes he was coming and just coming to my room, and we were just chatting. Yes, there is a possibility. I mean, for myself, always my physically the door is open. My heart also is open. We can always make a sharing and discussing, producing, and as you know, we know each other very recently. I'm very happy about the collaboration with you, and hopefully we will increase this kind of relationship with the
Sure,
other
sure.
scientists as well.
Thank you, Professor Ortas. Well, friends, we had the lecture of a real scholar and a scholar of real eminence. And Professor Ortas, I can assure you, all of us have thoroughly enjoyed. It's simply a fascinating stuff, and we pray God that you continue with this style of working and keep enlightening us with your excellent knowledge, not only in India but worldwide. May God bless you, and thank you so much on the behalf of Indian Society of Horticulture Research and Development, and on behalf of Dr. S.K. Dwivedi, our friend Mr. Seva. We profusely thank you for spending your such a precious time. Today happens to be Sunday, and we are so thankful to you. Thank you so much, Professor Ortas. Thank you.
Thanks. I was very happy to talk to Indian scientists and India's horticulture organization, and also thanks to you, and to continue our relationship, and hopefully to see in the next years in International World Citrus Congress in
Sure,
Adana.
sure.
We are really be happy to see you in Turkey and Indian scientists, horticulturists. Since I am in that organization, I will pass the information to you, and we will be happy to collaborate.
It will be a pleasure.
Thanks a lot. Thanks a lot.
Thank you. Thank you.
Yes. Okay. Cheers.
Thank you so much. Cheers.
Thank you very much.
Thank you so much, sir.
Thank you.
So much, sir.
Thank you.
Thank you.
Say hi to the professor. What's